Welcome to biotechnology. First of all, we should probably define it. Biotechnology is when we alter biology to get it to do things that we want it to do, rather than the things it would normally do. So the simplest case of that, or the most original case of that, would be farming. If we farm things for a long time, choosing the ones that give us more suitable crops, so oats, barley, corn, maize in South America, rice. These crops have been with us for a long time, for thousands of years, and we've altered them over time by choosing the ones that are more suited to our use of them, easier to digest, all kinds of things. We've managed to get potatoes up to a starch quantity of more than 99%. So we can get crops that are very effective just by choosing choosing the children of the plants that do the best. Livestock is much the same. So dogs we've altered from wolves, cats, their size and tendencies to do things are altered, although they're not as successful as dogs, and cows, which cows particularly are completely different size, completely almost helpless without us. The very large milk cows struggle to give birth naturally. They are mostly given cesarean section to deliver their calves, because they are just not the right size. They're not really suited to life anymore. We've altered them so much that they really can't survive on their own. In our case, we're going to take some bacteria and we're going to genetically alter them using a higher form of technology. So we're going to interfere with their genes to get them to make a protein that they normally wouldn't make. So the bacteria isn't able to make the protein, it doesn't do that, it has no use for it, but we alter its genes, we alter its genetic material and get it to do that. We'll do that later this semester, assuming that it all works. The way that I normally think of it is that we see things in biology either for whole organisms or just or simple chemical motifs, and we use them for other purposes. So in the case of a virus test, for example, we are using completely normal processes where animals recognize different things, and we use that to our advantage. So for example, in a pregnancy test, we are using the immune response of an animal to something, and generally we're using its immune response to measure a human protein that it normally would never be in contact with. Typically we would take a rabbit or a goat or something, or a rat, and we would inject it with a human protein, which is called HCG, human chorionic gonadotrophin, and we would generate some antibodies to it, and we would use those antibodies in our urine test to detect exactly that protein. And in the natural world, that would never happen. A mouse is not going to be injected with human wee at any point, so that's never going to happen. But we can use it to do that kind of thing. Obviously, we're not injecting them with human wee. We're injecting them with purified protein and collecting the antibodies. We don't need to do that directly anymore, but that is nominally what we're doing. The antibody response is very useful to us because then we can recognize all kinds of molecules, and that is one of the major parts of biotechnology. Another part is getting other proteins that we might want to make. First of all, we need to know more about genetics and where all of these proteins and other compounds come from. So now we'll get on to our topics. So I thought I'd try it a little bit differently this year. So we go straight into genes. So if we take a piece of genetic material, that would be some DNA. I keep saying so, I know I do that a lot. I'm trying to avoid it. So let us take some DNA. it contains a whole lot of bases, as we all know, and it's spiral. It's a double helix, as you will also know. What is more interesting for us is that the data contained on it that will be converted into a protein is held in 3 base codes known as codons, and they have so they're translated into amino acids. What you may know is that there are 20 amino acids, there are 4 bases, and so that's why we need to use more than one base to code for enough different codes. There are, because we're in 3s, we actually have a lot of redundancy in here, so we have extra codes that we can use, or that animals and plants can use. A lot of them code for the same amino acids, which allows it to be a little bit more resistant to error. And there are also special extra codes for starting a protein, for example, and stopping it. And we can use them for extra amino acids should we want to. So there are some researchers who've changed the code or changed the usage of some of these pieces of information to generate new proteins that contain amino acids that don't occur in nature, or aren't used in proteins in nature, better said. So let us get to the more important bit, the start. We need to make protein from this DNA, and so to start with, the DNA needs to be converted into some form that can be turned into DNA. So, sorry, let's try again. The DNA is eventually converted, the message inside the DNA is eventually converted into protein. Let us have a look at how that happens. So the easier way to do this is to look at bacteria, because they are comparatively simple. They don't chop around their DNA quite so much as we do. So in a bacterium, we would have a region that denotes where to start copying, and we would use that to make some RNA. So this is DNA. DNA is double-stranded. It is converted into RNA from this region along. So this is copied into RNA, and that will become the template for making proteins. So we have our RNA bases on here, and they match the DNA bases. We will think about the copying process in a moment. So this would be RNA. This type of RNA is messenger RNA, so it's got a little M. So our messenger RNA now needs to, or now is used to make protein. So our messenger RNA has its codons on there, which are 3 base pairs, and RNA has only a single strand. It doesn't pair up like DNA. So these all stick out, and that is used in a way that you may have seen in school. So this piece of tRNA brings along an amino acid, and they all eventually, a piece of tRNA brings along a specific amino acid, reasonably specific anyway, and they join onto each other to form the protein backbone, effectively. And that is assisted by some cellular machinery which uses the starting part, which is why I've left a bit at the beginning, which uses the starting part to initiate the process. One piece of RNA, one piece of messenger RNA, that is, generates several molecules of protein. It can be a lot of molecules of protein, but eventually it is broken down, and that's useful because a cell, like our cells, or these in the case of bacterial cells, will need during its life to turn on and off protein production several times, and if it makes messenger RNA and then it will produce protein forever, that would mean it would be more complicated. So it has, as very often is the case in biological processes, they have a sort of best before date, they run out gradually, and so there is, unless it's being generated continuously, the amount of protein production will go down and get down as it gets broken back down again. That means that the body only needs to turn this on, or the cell only needs to turn on the production of messenger RNA and turn it back off again in order to turn on and off the production of whatever the protein is, although the response time of the cell is not super fast because the messenger RNA is still there. If it needs to stop it more quickly, it would have to do something to interfere with this process, which is also possible. Okay, so now it's been a little while for me, not so long for you, and I want to see how DNA might be copied. So first of all, we have to separate the two strands, or that's what happens in DNA copying. And the next strand of DNA will grow in one direction on one side and in the other direction on the other side. It could grow in both directions, but it turns out naturally there's only one direction that it tends to grow in, and that's because there is an enzyme that does that. And it works in one particular direction because, I should have told you that, because DNA is directional. So we've got our DNA in one direction and our DNA in the other direction on the other side. This is a speciality of the types of bonding that we have. So effectively we've got a 5-membered ring. Let's draw the 5-membered ring. Oops, badly. Draw the 5-membered ring. We've got a 5-membered ring, which means there's a directionality to the process, to the strand, and that informs the direction of the movement, of the growth. This is significant because it means that we can't grow DNA easily in the other direction, so in this direction or in this direction. It's also significant because the two strands are effectively differently coded, because this one is going in this direction, the other one is going in the other direction. So the enzyme that causes this to grow is known as polymerase. The enzyme that helps this to grow is called polymerase. If it's DNA, then it's DNA polymerase. And if we assume that this is the strand that contains the information, the other one is going in the backwards direction, so it doesn't contain the information that we want, it could be RNA polymerase, which is going to copy just the one side. If we need to copy the whole strand of DNA, which is usually the case, then we will have a problem when we get to the next bit, or we'll have several problems. The first problem is it will take a really, really long time to copy one base by base all the way along to the end of the chain. Then we'd end up with two completely separate chains. But what happens backwards of this point? And if we assume that this happens at several points along our DNA, then we'll end up connecting several at the end, which means that we need a new enzyme when we get to the next one. So let's draw that. Maybe that's a good idea. So this is the next one coming back. And when this one reaches it, let's get rid of that strand over there, give it a bit of space, then we will have to connect the two together, and that's done by a different enzyme that's called ligase, which glues up this last connection. We need that because later on we'll be using that enzyme for other purposes. For the mRNA, yes, we will not need to do that. We just need to make an RNA strand along here, which was in red before. So let's make that red instead. If it's an RNA, then we need to just copy some of our DNA and send it for production of enzymes or proteins, whatever is going to be made. This brings us to our first significant technology, which will use this enzyme, DNA polymerase, which has, DNA polymerase, which has the function of growing DNA, but it can only do so if it's got a start. So we've got our piece of DNA that we would like to copy. It's either a piece of DNA that we would like to copy because we want to use it to make something, or it's from a crime scene and we want to copy it because we want more to analyze, or whatever we want it for. Or maybe we want to just find out whether it's the DNA of corona, and so we need to copy it to make more so that we can compare it to something else. So we would like this DNA, we would like more of it. That's our job. Now we've got to have a functional analog of opening that crack in the DNA, and that's normally done in the natural system with enzymes and proteins, helper molecules, and we don't have those. So we're going to heat it up. So let's do that. Delta H, we have to heat it up to 90 degrees, which is quite hot, and it falls apart into two parts. Why does it do this? Well, we've got a standard reaction. Effectively, we've got our Gibbs free energy, which is equal to the sum of the bonding. So we get bonding energy out, maybe, in one direction. And we have a problem with entropy in the other direction. So if we heat it up, we have two strands. There's more going on. If we have the DNA in its normal state, there are hydrogen bonds between all of these intermediate groups, the bases of the DNA strands, and they hold them both together, enabling it to be relatively stable. Each hydrogen bond is not very stable, particularly in water, because the water can come along and interfere. It can bond to either end of the DNA hydrogen bond, so either the OH or the nitrogen part. So it can mess with this hydrogen bonding structure and make it fall apart, but the probability of it being able to do so all along is very low unless we increase the temperature. If we increase the temperature, the advantage of having more parts is higher, so we can separate it into two strands. That's good, because if we heat it up, so this is about 90 degrees C, oops, it's hard to write from the side, that's why my handwriting is so shitty. And it's also too low. Let's put it on up here. So it's about 90 degrees C. They fall apart into single-stranded DNA. One of them being the sense strand, the one that contains all the information that we want, and one of them being called the anti-sense strand. It's the opposite of the sense strand, and it's not useful directly, but obviously if we copy it, we can get somewhere. Now we want to get started. We've got a physical way of getting the opening up mechanism. We can also have a dodge for the getting started mechanism. So instead of having all of the proteins and cell mechanisms, cell machinery to get the process started, we can do it in a different way. And we're going to do it this way using a thing called primers. So these are little sections of DNA, DNA, single-stranded DNA, which will stick on the beginning and the end. And they will have quite a lot of bases. I've only drawn a short bit because my DNA is non-realistically short, but they're a lot longer than this. Their job is to recognize the DNA, and they have a massive advantage. So if we're doing a coronavirus test, for example, this would be specific to the type of virus that we're looking for, the coronavirus family. There's another complication, but we'll get into that later. But we're looking for a specific match to some of our DNA, and this will copy from here forwards and from here forwards, which I've just decided is going to be downwards and upwards in this direction. It's always in the opposite direction. Just as before, the polymerase only works in one direction, so anything that is on the other side of this, imagining this strand is longer in this direction, anything that's on the other side of this will not get copied. That has also in relatively far-reaching consequences. If our DNA is very long, we're only going to copy a section. That can be useful If we want to use it to identify a virus, we are only interested in the small section to check that it's the same thing. If it's the whole genetic material of somebody and we want to know, we want to copy it all, that is not so easy with this method. If we copy this section, we're going to copy it many times. So as we cool it down, I should have changed my number here. At 90 degrees C, they're separate. As we cool down, the primers attach. So I'll take my numbers off. As we cool it down, the primers attach. And as we cool it down further, let me write primer. So this is the primer. As we cool it down further, the DNA polymerase gets into action and finishes, I've got another color, and finishes the rest of the strand, leaving us with 2 new strands of DNA, which we can run through another cycle. The good thing about this reaction is all we have to do is heat it up and cool it down, and every time we get a doubling, which is the basis for this controversy that we have at the moment for the PCR test for coronavirus, that it's done particularly many times. So we can heat it up and cool it down. Every time we do that, we double the amount of DNA, but only in the section between the 2 primers. And only if we've got plenty of primers and plenty of bases in solution, so that it's not hindered by any of those components running out. Obviously, if they run out, we can't make any more. There is some special DNA left over. The original strand of DNA is left over as this tail on one of the pieces of DNA, and this one, possibly a tail on the end of this one, and up here, this is going to copy this extra bit, so the original DNA was here, so this is the first one will be longer, and the first one will be longer here, so there'll be some families in our pot that are not the same. Most of our DNA will just be the bit between the forward and the reverse primer, because every time we double, we're ending up with a lot of molecules, and most of them will just be that intermediate region. There will be some left over. That's the starting bit, and there possibly, if it's a crime scene, will be a whole lot left over that is not from not matching the primer. So if we imagine it's a crime scene and we are looking for human DNA because we imagine that the human has done the murder or whatever it is, then we deliberately cutting out the cellulose from paper, because cellulose hasn't got any DNA, but the tree does. If we do an extraction of cellulose and we use the wrong primer, if we do an extraction, we use paper and we use the wrong primer, we're going to end up with a lot of DNA that's related to effectively evergreen trees. If we do, alternatively, there will be a lot of bacteria, a lot of fungus, all kinds of organisms in our sample, and we want to focus on one particular thing. Likewise, in a corona test done on a human, we've taken some material, so it's not effectively some horrible gunk, out of the nose of a human being. There is mostly human cells in there, mostly human DNA, and we don't want that. We want the virus DNA, and we don't just want any old virus DNA, we want a specific type of virus DNA. So then we choose the primers so that they copy mostly that and not of anything else. That's good. It makes it a very powerful technique, because then we can ignore lots of background information that we don't want. The problem of just doubling everything would be that we would have a lot of other things. We would not be improving our situation. Every copy, and here we are. We're selecting one family or one smaller range of DNA. In the case of a crime scene, just human. In the case of some of the coronavirus tests, just Corona-19, the one that is causing our pandemic, but or in the case of others, all coronaviruses, so we can also test for flu and colds and things at the same time. We would then have to have another step to work out which one it was. What matters is what what the exact primers are and whether we have two or whether we put more in to copy some other DNA. Physical process that we have to do is just warm it up and cool it down. And the reason that the temperature is so high is because we use DNA polymerase from a type of bacteria that live in a very hot place in undersea vents, so that it can take very high temperatures, and that makes it more, well, it makes it quicker, and it makes it more resistant to the temperatures that we have to heat it up to get the DNA to turn into monomers, to turn into single-strand DNA without having any other cellular material in there, which is good because then the less things that we put in, the cheaper it is. Here we've got one enzyme, two primers, and the bases, and that's it.